Hi everybody, and welcome to the Oxygen Content and Air Lab at Tacoma Community College. The main goal of this lab is to figure out how much oxygen is in air versus our exhaled breath. And if you think about air, just the standard air in our room or in our environment, it's made up of a variety of gases. The main gas is called nitrogen. It's actually a very small portion of the air around us that's made up of oxygen. But during today's experiment, we'll be able to figure out exactly what percentage of oxygen makes up the air around us. And it's interesting, if you think about this oxygen, it's the only gas in our air that can rust with iron. So what we're going to do today is we're going to stick some steel wool inside of a syringe, and that steel wool has iron in it. That iron will react with the oxygen in our air or in our exhaled breath and form rust. When this reaction takes place, if we have excess steel wool, all of that oxygen in theory could be consumed and converted into iron oxide, which is rust, meaning there would not be any gaseous oxygen left inside of that syringe. From that point, we can divide the amount of oxygen that was consumed by the total volume of air or exhaled breath on the inside of the syringe to find the percentage of oxygen making up that sample. So let's go ahead and jump into the video and see how this experiment works. The experimental setup is pretty straightforward. You'll want two beakers, and you'll want to fill them approximately half full with water. I've got one labeled as breath, and then the other one labeled as air. I've got two timers, one for each beaker, and I've got things pretty well labeled, that way I don't mix them up later on. Additionally, I'm going to grab some dish soap, and I'm going to put a small squirt into each beaker to help minimize the effects of the meniscus on the inside of the syringe. That will make it easier for measuring. In addition, in this small beaker, I'm going to add about 30 milliliters of vinegar. This is just standard vinegar that you have in your kitchen, and this will help clean the steel wool and allow it to rust at a much faster rate. So let's measure out 30 milliliters. And then, to further help the rusting process go faster, I'm going to add a few scoops of sodium chloride. This is just standard table salt that you have in your kitchen. So I'll go ahead and add a couple scoops into this, and we'll use this solution to rinse our steel wool. It's a good idea to try to mix this up with the glass stir rod, so remember to do that before you mix your steel wool in. It'll help the rusting process go much more smoothly during your actual experiment. Next, I'll take my vinegar solution over to the sink and submerge my steel wool for about a minute to clean it. It's a good idea to kind of swirl it around in there, and if you need to use your tweezers, you can poke it down a little bit to ensure that it's fully saturated. Be sure to let it sit for an entire minute. If you let it go for too short, you won't clean your steel wool thoroughly. Alright, after about 1 minute, what you want to do is you want to grab your tweezers. Then grab the steel wool, take it out of your beaker, shake off any excess vinegar salt solution, and then you want to open your syringe, and you want to put your steel wool in till you get to about 25 milliliters. You want that steel wool to be nice and high in your syringe, not too low. You'll also notice there's a hole there. So when you push your syringe top back in, make sure that the hole isn't open anymore. You want the plunger to be down past that hole. Now we'll take it back to our fume hood. And I like taking off the little rubber piece on the bottom, and I'll use my clamp to hold this in the solution so that the tip is submerged. After I have it submerged, I'm going to suck up just a little bit of solution so that it's starting exactly at 0.0 milliliters. Once it's set there, I'm going to start my timer. Make sure that you also measure where the bottom of your plunger is. You want to make sure that it's measured to the proper number of significant figures, and you want to include that volume in your lab notebook. I'll include this volume in the lab handout so that you have it for your reference. For the second syringe, we want to do something similar. We'll grab our steel wool, shake off any excess solution after it's been sitting in there for 1 minute. We'll open up our syringe, and then gently place our steel wool in the top portion of the syringe. After we've got it down at the appropriate height, we'll add the plunger back in, and then we'll take this syringe back to our fume hood. Inside of my fume hood, I have a balloon full of exhaled air. The clamp is used to close this balloon, and so I'll just quickly take this clamp off, and then I'll put the balloon over the black tip and then I'll pull the plunger back so that that hole at the top of the syringe is open. Once that hole is open, I'll let the balloon release so that the air inside the balloon, meaning my exhaled air, flushes all the way through the syringe and out the hole at the top. The purpose of this is to push out any air from the room. We want the inside contents of the syringe to only be exhaled air, which is why we're using a pretty big balloon here. It may take a minute or two for this air to go through, but just be patient. When you're done, push the syringe back down so that the hole is no longer open. This is very important, otherwise room air will get back in. Just like before, I took off the black tip. I'll put it clamped in my hood, and I'll ensure that the tip is submerged, and I want to pull up just enough water so that it starts at exactly 0.0 milliliters. And then I'll hit start. In addition, remember to measure the exact volume for the top of your plunger. Now, lucky for you, you don't have to sit here for the next hour collecting data. Instead, I've done that for you. All of the data can be found in your lab handout. Make sure that you answer all of the questions as described in your lab handout and submit your lab report as described by your instructor. If you have any questions, feel free to contact your instructor or a tutor at the Writing and Tutoring Center. Good luck!